Can you imagine a world where tuberculosis, one of the deadliest diseases, can be diagnosed with the precision of an expert in just minutes, all thanks to artificial intelligence? Today on AMED Revolution, we explore a multi-center study that could change the way we treat infectious diseases in resource-limited settings. AI is no longer just a support tool. It could be leading the way in clinical diagnosis. Stay tuned to discover how this advancement promises to improve the lives of millions of people worldwide. About 30% to 40% of tuberculosis, TB cases go undiagnosed, contributing to the transmission and worsening of the disease, especially in low and middle-income countries. This severely impacts the achievement of the global end TB strategy goals set by the World Health Organization, WHO, which aims to eradicate tuberculosis by 2035. The issue lies in the lack of access to affordable, accurate diagnostic tools. In many of these countries, molecular diagnostic tests like GeneXpert are costly and not readily available. Although chest X-rays, CXR, are more accessible, interpreting these images requires experts who, in many cases, are not available. This leads to delays in diagnosis and, consequently, late treatment. In response to this situation, in 2021, the WHO recommended the use of computer-aided detection CAD software to automate chest X-ray readings and improve TB detection in resource-limited settings. However, more research has been needed to validate the performance of these tools, especially in populations with a high burden of HIV, where chest X-rays can be more challenging to interpret due to atypical pulmonary presentations. This study, published in New England Journal of Medicine AI, aims to fill this knowledge gap by prospectively validating two artificial intelligence models in Zambia, a country with a high prevalence of TB and HIV. The study evaluates not only an AI model for TB detection in chest X-rays, but also a secondary model that detects more general pulmonary abnormalities, which could help improve the diagnosis of various lung diseases. The study was conducted between November 2021 and February 2023 at 3 clinics located in the Lusaka district of Zambia. These clinics, named Kanyama, Chawama, and Chunda, are specialized centers for diagnosing and treating tuberculosis. A total of 1,978 adult patients were recruited, all of whom either exhibited symptoms of tuberculosis, cough, fever, weight loss, or night sweats, were close contacts of confirmed TB patients, or had recently been diagnosed with HIV. This approach was crucial as it allowed the evaluation of AI performance in a population at high risk of active TB and HIV co-infection, adding complexity to the diagnosis. The study process included several key steps to ensure the collection of high-quality data. Each patient underwent a chest X-ray, CXR, and a detailed clinical evaluation that included medical history, physical examination, and sputum sample collection for microbiological testing. The CXR images were anonymized and uploaded to a cloud system using Google Cloud Digital Imaging and Communications and Medicine platform. Subsequently, the two AI systems, one for TB and another for pulmonary abnormalities, processed the images within minutes through an automated pipeline. It is important to note that, since the study was observational, the AI results were not used for clinical decision-making during patient visits. TB AI model. The AI model for TB was trained to identify patterns in chest X-rays that match signs of active tuberculosis. The AI generated a continuous score from 0 to 1, which was then converted into a binary result, positive or negative, using two predefined thresholds. The first was a high sensitivity threshold designed to maximize TB case detection, and the second, a more balanced threshold that mirrored radiologist performance. Pulmonary Abnormalities AI model. This second model evaluated the presence of a wide range of pulmonary abnormalities, such as focal consolidations, pleural effusions, and lung masses, aiming to be used as a triage tool to prioritize the diagnosis of respiratory diseases. Patients' sputum samples were sent to central laboratories for analysis. Tests such as Expert Ultra, a molecular technique, and Mycobacterium tuberculosis culture were used as the reference standard for determining the presence or absence of active TB. For the evaluation of the Pulmonary Abnormalities AI model, the images were reviewed by 3 independent radiologists. The radiologists, who were blinded to the clinical data, mark the presence or absence of abnormalities and the specific types observed in the images. The study successfully analyzed 1,827 patients for the TB-AI model and 1,910 for the pulmonary abnormalities AI model. Here are the most relevant findings. The study successfully analyzed 1,827 patients for the TB-AI model and 1,910 for the pulmonary abnormalities AI model. Here are the most relevant findings. Pulmonary abnormalities AI model performance. This model achieved a sensitivity of 97% and a specificity of 79%, meeting the study's predefined targets of 90% sensitivity and 50% specificity. This means that AI is highly capable of identifying a wide range of pulmonary abnormalities, making it a valuable support tool for physicians working in areas without access to radiologists. These results are especially relevant in the context of a global pandemic, where respiratory infections have increased the workload of healthcare systems. The ability to prioritize abnormal chest X-rays using AI can significantly reduce waiting times for diagnosis. The study also explored how the performance of AI and radiologists varied across different patient subgroups. Specific analyses were conducted based on HIV status, previous TB history, age, and sex. In the HIV-positive subgroup, both AI and radiologists performed worse compared to HIV-negative patients. AI achieved a sensitivity of 82% and a specificity of 65% in this group. This is because HIV-positive patients often present atypical chest X-ray features, making accurate TB detection more challenging. For radiologists, the sensitivity was even lower, averaging 66%, suggesting that AI could be particularly useful in populations with a high prevalence of HIV. Patients who had previously had TB also posed a challenge. AI showed an AUC, area under the curve, of 0.76 in this subgroup, indicating a drop in accuracy compared to patients with no history of TB. Radiologists also perform worse in these cases, with a significant drop in specificity. This finding highlights the importance of adjusting AI thresholds for specific populations, something the study suggests as a key area for future research. In terms of age, both AI and radiologists perform worse in patients aged 40 to 65, which was partly attributed to the correlation with HIV status and prior TB. Overall, AI and radiologists showed better results in younger patients. Regarding sex, AI's performance was slightly better in men than in women. This could be due to the higher prevalence of TB in men within the study cohort. The study highlighted several important conclusions worth exploring in more detail. While the TB AI model did not achieve the sensitivity recommended by the WHO, it outperformed radiologists in most cases. This raises questions about whether the WHO's goals are realistic in populations with a high burden of HIV, where pulmonary patterns may not be typical. The fact that AI achieved higher sensitivity than radiologists suggests that, with further adjustments and calibration for local populations, AI could improve even more and potentially meet WHO goals. The use of AI to detect abnormalities in CXRS beyond TB has immense potential, especially during global health crises like the COVID-19 pandemic. The high sensitivity of AI in identifying abnormalities can reduce the workload of radiologists and enable faster intervention in urgent cases. Additionally, AI offers advantages in terms of cost and accessibility. Unlike radiologists, who may not be available in rural or resource-limited areas, AI can run on the cloud or on local devices with very short processing times. A major limitation was the use of a single shared X-ray device between two of the study sites, which introduced artifacts that could have affected AI performance. However, despite these issues, the AI models performed well, suggesting inherent robustness in their design. Another limitation was the exclusion of certain patients due to the lack of a valid reference standard, e.g. contaminated samples, which could have influenced the final analysis results. The future of medical diagnosis is here, and artificial intelligence is playing a key role. This study shows that AI can not only match the performance of radiologists in resource-limited settings, but it can even surpass them in detecting tuberculosis and other pulmonary diseases. In a world where millions of people go undiagnosed in time, AI offers a real hope of improving access to healthcare. If you're passionate about the intersection of technology and medicine, don't forget to subscribe to AMED Revolution and continue exploring with us how artificial intelligence is shaping the future of healthcare. What do you think about the use of AI in clinical diagnostics? Share your thoughts in the comments and let us know your ideas.